Hello everybody. We are going to talk about monohybrids today. So we briefly talked about Punnett squares and using them to predict possible outcomes of offsprings. So now we're going to do some additional practice today, looking again at monohybrids, mono meaning one. So we're looking at how one trait can be predicted in some different offsprings. How I would like for you to use this today is in conjunction with the worksheet that you have on iTunes U. So go into iTunes U, and this is the Crazy Critters Part 2. So you're going to look at that and you're going to follow along. As you are listening to me, I'm sorry you've got to listen to my voice today because I'm not there, but you have an opportunity to listen and watch how to do the Punnett squares. And if you're not sure what to do, you can go back and actually re-listen. So don't just follow along, fill in. What I would like for you to do is actually try to work the problem and then listen to my solution. So what I need for you to make sure that you have open, it says right here, it says use the genotypes and phenotypes using the first part of this activity. So if you open up that activity and go to Table 2, key to genotypes and phenotypes, that will help you determine which of these traits are dominant, which of these traits are recessive. So if we look at this first one, the first one says we've got a female crazy critter with taloned feet that was bred with a male that is homozygous dominant for her feet. Sorry, for his feet. So first we've got to look at taloned feet, or just the feet in general. So if we go there and we look at feet, or the foot, that's trait number 4. So the dominant trait is webbed, so taloned feet would be little t, little t. So here, with the taloned feet, our female genotype is going to be little t, little t. Now we've got to look at the male. The male, it says homozygous dominant for feet. Dominant, making sure that we use the capital letters. And so homozygous, homo means one or the same. Homozygous dominant for feet. So it's going to be big T, big T. So now we need to move those letters down to the actual Punnett square. It doesn't matter if you put the female on the top or the side. You're going to come out with the same offspring options. So we're going to go ahead and put little t and little t at the top, big T and big T on the sides. So each one of our offspring are now going to be big T, little t, big T, little t. So what does that tell us? That means if we were to have children and we had one crazy critter that had taloned feet and one that was homozygous dominant for feet, all of their offspring would be heterozygous for that trait. So now we know that we have 100% of those offspring are going to have that webbed foot. So it says, what is the genotypic ratio? Guys, remember genes are in your DNA. So the genotypic ratio, we still have to look at all 3 possible genotypes. So the first genotype, we normally start with homozygous dominant. So we have 0, big T, big T. We have 4, big T, little t. And we have 0, little t, little t. So that's our genotypic ratio. Phenotypic ratio is going to be a little bit different. If the offspring have, or even the adults have webbed feet, it's a dominant. So it's a completely dominant trait. So they could be big T, or big T, little t. So our 2 options are webbed feet and taloned feet. So because all of these in here, all 4 of these are going to have big T, little t. We're going to say that there are 4, and then that dominant trait there is webbed. And that's going to be 2. How many of them have taloned feet? Well, none of them are little t, so we're going to do 0. Alright, so we've got 0, and that phenotype, because we're looking for the phenotypic ratio, is 0 talon. Alright, then percent of offspring, so we've got 4 webbed, 0 talon, so we've got 100% that are going to be webbed. We have 0% that are going to be taloned. Let's try question number 2. If you feel like you have this down, go ahead and pause this, try it on your own, and then see what the solution is. If you still need more help, go ahead and watch me talk through this, and then you can always go back and listen to it again if you need to. So a female crazy critter with heterozygous genes for feet is bred with a male that is homozygous recessive for feet. So a female crazy critter that is with heterozygous genes, so that means it's going to be different. Hetero means different. So there we're going to have our big T and our little t. The male says it's homozygous recessive for feet. Homo again meaning same, recessive, little letters. So the male is going to have a little t, little t. Again, it doesn't matter where you actually put the letters. We'll put big T, little t there, and little t on the side. So remember, you're going to bring this down here and that over there. And so that's going to give you big T, little t. Do the same for the next one. Bring that t down, little t across. So again, we've got big T, little t. Here, we're going to bring this little t across, that little t down. So we've got little t, and finally the same thing on the last one. Little t across, little t down. So here we've got little t, little t. So the genotypic ratio, just like before, even if we don't see all 3 possibilities, we still need to write it. So this is a ratio. So here we've got none that are big T, so we're going to put 0 that are big T, and we're going to 2, so 0 big T. Here we actually have 2 that are big T, little t. And we've got 2, big T, little t, 2 of them that are little t, little t. Now the phenotypic ratio, again, is what you physically see. What are we seeing here? So if we go and we look at the feet again, we know that the feet, the big T, and the big T, little t are going to show webbing. So we've got 2 that are webbed, and we have 2 that are going to have the taloned feet. Now, just like in math, if you've got a 2 to 2 ratio, can you reduce that? Yes. So essentially we're saying it's 2 over 2, which we can reduce to 1 over 1. You can leave it 2 web to 2 talon, or you can reduce it 1 web to 1 talon. Either way, I will accept that as long as you can reduce it properly. So what percentage of the offspring will the taloned feet have? There we've got 50%, and webbed is 50%. All right, on question 3, once again, if you feel comfortable with this, go ahead, pause or stop the recording, go ahead and work through this, and then if you feel comfortable with it, then you can go ahead and work through it, listen to me afterwards, make sure you know what you're doing. If you're still not quite sure, go ahead and listen through this, make sure you know what you're doing. Alright, so a female crazy critter and a male crazy critter both have heterozygous genes for feet. Hetero meaning different. So Here we've got our female, so hetero different, big T, little t. The male, big T, little t. So these are both the same. We worked actually through this yesterday in class, so this should look familiar. So up here at the top, we're going to put big T, and big T, little t on the side. So in this first one right here, just like we did before, bring the t's down, and we're going to have big T, big T. The next one, big T, little t. We're going to go across, big T, and little t, little t. So our genotypic ratio, we now have all three different varieties. So here, the first one, we're going to always start with the dominant, the homozygous dominant. So we're going to just do 1, big T, 2, 2, big T, 2, 1, little t. So the phenotypic ratio, because again, this is a complete dominant trait, the dominant's going to always overshadow the recessive. The big T, and the big T, little t are both going to be webbed feet. So now we have 3 webbed, 2, 1 talon. So what percent of the offspring will have taloned feet? 25. And webbed will be 75. Okay, letter E says, how does this compare to the actual percentage in Table 3? So go back and look to see what you got. I know y'all didn't write the numbers down today, but you kind of remember what those percentages were, and kind of explain what did we talk about today in class, what were some of the numbers that you saw versus what were you actually getting here. All right, so now we're going to do a little bit different monohybrid. The first 3 that we did were complete dominance, dominant overshadowed recessive. Now we're going to look at incomplete dominance. So in the crazy critters, we looked at 2 different traits that were incomplete dominant. One was the skin color. It was either red, purple, or blue. And the second one was the tail length. It was either long, short, or it was absent. So remember, in incomplete dominance, your traits are going to blend. So you're going to have that nice mixing, kind of like a middle between your 2 different traits. So they're both actually dominant. So in the crazy critters activity, we still represented blue with little t. But there is no recessive trait in incomplete dominance because it's a dominant trait. So now we're going to fix that. We're going to call blue T1 and the red just letter T. So if you look here, right in here, you can see the little ones that are actually above the T's, and that's going to tell us the difference between the red and the blue. So here we says a female red crazy critter is bred with a blue male crazy critter. So the female is red, so she's going to be big T, big T. The male is blue, so he's going to be T1, T1. So again, we're not using little letters because there is no recessive trait in an incomplete dominant trait. They're both dominant. So we have to represent that with a capital letter. And we're showing that they're not the same trait. They are the same trait, it's just a different version of that trait with that 1 as that superscript, so that way we can tell them apart. So now we put them in the Punnett squared, just like we did before. We've got big T here, T1, T1 here. So just like we did before, we're going to put those into the Punnett square. So we're going to say T, T1, T, T1, T, T1, and T, T1. So if we have a red crazy critter bred with a blue crazy critter, then all of their offspring are going to be purple. We do the genotypic ratio and the phenotypic ratio pretty much just like we did before, but we'll talk about a little bit of the difference with that phenotypic ratio. So the genotypic ratio, we still need to show all three possibilities. So we have 0, big T, 2, sorry, let's make that a little bit clearer. I made my dots align. There we go. So 0, big T, 2, we've got 1, 2, 3, 4, big T1s, 2, 0, T1, T1. Okay? The phenotypic ratio, again, we need to write all three, and I'm going to kind of run out of room because I'm trying to write this on the screen, so bear with me. We've got to put all three. So we have 0, red, to 4, purple, to 0, blue. Look at that, I made it. So it's a little bit different because we've got to put all three phenotypes. And then the percentage is just like we've done before. So there was 0% red, 100% purple, and 0% blue. Alright, so in question number 5, we've got 2 purple critters that are going to breed together. So the female is purple and the male is purple. So on female, we're going to have big T1, and the male will be big T1. So again, just like before, we've got big T1 at the top, big T1 on the side. First box, we're going to have big T, down below that, big T1, over to the side, big T1, and the last one is big T1. So the genotypic ratio, we actually have all three representatives. So we're going to have 1, big T, 2, big T1s, 2, 1, T1, T1. Phenotypic ratio, again, we have all three representatives. So we have 1 red, to 2 purple, to 1 blue. Percent of the offspring, we've got 25% red, 50% purple, and we have 25% blue. So again, it asks you to compare this to the actual percentage in table 3. Again, y'all didn't write it down, but think about what we talked about yesterday and how does that compare to what the class actually got for our data. Alright, so this next one that we're going to look at is called codominant. We talked about that one yesterday. Codominant in those crazy critters was where you had the red and the green eyes. So remember, co means equal. So when you have like a co-captain, they have equal responsibilities. So in your codominant traits, you're going to see those traits equally. So here we have a red-eyed female crazy critter bred with a green-eyed male crazy critter. So red, just like before, we have no lowercase letters. So instead, for the little letters that we used in the lab yesterday, we're going to use the T1s just like we did before with incomplete dominant traits. So here, for female genotype, we're going to do big T. For the male, because the male is green-eyed, we're going to do T1 and T1. Now we set it up exactly the same. We're going to put big T's on the top, T1s on the side, and then all of the offspring will be T1. Alright, so the genotypic ratio, again, like we've done before, we need to show all three possibilities. So we have 0 that are big T. We have 4 that are big T1, and we have 0 that are T1. So the phenotypic ratio again, like we did before, we need to show all three possibilities. Again, I might run out of room, so just be patient. So we've got 0 that are red, to 4 that are red and green, to 0 that are green. Look at that, I ran out of room on this one. Okay, a little green over there. So we've got 0 red to 4 red and green to 0 green. So on our percentages, pretty easy. We've got 0% red, we have 100% green, and 0%, sorry, it was 100% red green and 0% just plain green. All right, the last one that we're going to do today is number 7. So again, this is co-dominant. If you feel completely comfortable, like I've said before, pause me, go through, fill it out, and then check your answers. So that's up to you. If you still want to listen, go through and listen. If not, work through the problem and then check your answers. So we have a red and green-eyed female bred with a red and green-eyed male crazy critter. So our genotype for our female is T1, male is T1. All right, so again, just like before, we're going to put the T's on top, T1 there. All right, so in this top box, we've got big T. Right below that, big T1. Over here next to the top one, T1. And then the bottom one, we've got T1. So our genotypic ratio, again, we want to show all three. So on this one here, we have 1 that is big T. We have 2 that are T1. We have this 1 that is a T1. Okay, so phenotypic ratio, again, I might write on a room. So here, your genotypic ratio, we've got 1 that is red, 2, oops, I didn't put my numbers up there at the top. I apologize. Let me go back. Genotypic ratio, 1, big T. We had the 2 T1s to the 1 T1. All right, let's look back at phenotypic ratio. We had the 1 red. We've got the 2 that are red-green. And then the last one, we've got the 1 that is green. Made it. Percentage, we've got 25% red. We have 50% red and green. Look at that, I forgot my number up there again. 2, there we go. All right, and then the green one, we've got 25%. Again, he asked you to compare this to the actual percentage in Table 3. We did that in class yesterday. We didn't write anything down. That's okay. Try to remember what the class data was about what we had on that one, and then go ahead and answer that question. This is the last one we're going to do here for monohybrids. Go back through this if you're still having problems. Go back through this. Listen to me again. I know. Listen to it again until you get this down. Because the next one that we do are going to be dihybrids, and they're going to be a little bit more challenging. So make sure you understand how to do the monohybrids and the difference between incomplete and complete dominance. All right. Have fun, guys.